But when you remove the carbonyl from uridine with the anti-cancer compound zebularine, which is identical, except it just doesn't have that 4-carbonyl group, we see a modest loss of binding affinity to 120 micromolar. And we saw the crystal structure of VCC&T bound to zebularine, and shown here is FOFC simulated annealing omit density for the zebularine, and the rest of the electron density that I'm going to show in the subsequent figures is also going to be that same type of electron density. So what we saw in the structure is this interaction is no longer there, and glutamate 156 actually adopts a different rotamer position, probably as a result of removing that interaction. In the uridine-bound structure, we saw this 3-nitrogen group. It formed a water-mediated interaction with glutamate 156 and threonine 155. If you block that interaction by putting a methyl group there with 3-methyluridine, we see a pretty significant loss of binding affinity. Similarly, with the bulky purine ring of adenosine, we saw a similar loss of binding affinity to about 470 micromolar. We saw the crystal structure bound to adenosine and found that there's no longer that water molecule here interacting with the nucleoside in the protein. And so the bulky purine base disrupts, or it sterically interferes with where that water molecule would have been, but it's not close enough to interact directly with these residues. Additionally, we measured the binding affinity of the antiviral compound ribavirin, which is a purine analog, and found it to be significantly poorer than adenosine and methyluridine, and saw the crystal structure bound to ribavirin and saw that it can't form any of these interactions either. Looking at the 5 position of the uracil base, there are several compounds that have substituents at this position, including the anti-cancer drug 5-fluorouridine. So we measured the binding affinity for 5-fluorouridine to be about 16 micromolar, which is about 2-fold better than for uridine. So we saw the crystal structure bound to 5-fluorouridine in hopes of getting a better idea of where this enhanced binding affinity is coming from. And although we see clear density for the fluorine substituent here, it doesn't form any new interactions within the binding site, but it's small enough that it doesn't disrupt the structure of the binding site itself. So we measured the binding affinities for a few other 5 substituted uridines and found that small, highly electronegative substituents at that position resulted in about a 2-fold better binding affinity, while bulkier, less electronegative substituents result in a loss of binding affinity. So what we think this is coming from the interaction of the pyrimidine base with phenylalanine 366, which sits there under the pyrimidine base. And looking a little bit more closely at this interaction, you'd see that it kind of sits directly under the pyrimidine base and forms a kind of pi-pi interaction. It's not quite parallel and it's not quite T-shaped, but the pi electrons will certainly be interacting. And we think that putting a small, highly electronegative substituent here is going to redistribute the electron density around the pyrimidine base and strengthen this interaction. Notably, phenylalanine 366 is the only residue within the binding site to interact with both the nucleobase moiety and the ribose moiety of the nucleoside. It interacts with the ribose via CH-pi interactions. And what we think it does is it serves to orient the nucleoside. The nucleoside comes in and sits on phenylalanine 366 and serves to orient the nucleoside so that it can form all the other interactions that decorate the binding site. Prior to our studies, phenylalanine 366 had not been identified as a residue that was important for nucleoside recognition, so we mutated it to alanine, tyrosine, and tryptophan and performed ITC measurements with uridine and saw a significant loss of binding affinity, really highlighting its importance in nucleoside recognition. So we have thus dubbed it the selectivity ring of the transporter. Now looking at the ribose interactions, we wanted to probe all of the interactions with the hydroxyls of the ribose by measuring binding affinities for uridines from which the hydroxyls have been removed. So we measured the KD for 2' deoxyuridine to be 170 micromolar, which is a modest loss of binding affinity, which is kind of expected since 2' deoxynucleosides were previously known to be permeates of human CNTs. We also measured KDs for both 3' and 5' deoxyuridines and found them to be almost immeasurable in the 3 millimolar range. And that also makes sense since those two positions are invariant among naturally occurring nucleosides. Looking a little bit more closely at that 2' position of the ribose, we measured the KD for cytidine to be 61 micromolar, but the cytidine analog, gemcitabine, which is identical in structure to cytidine except it has 2 fluorine atoms bound here at the 2' position, exhibited a significant loss of binding affinity to over a millimolar. And then furthermore, the cytidine analog, cytarabine, which is an anti-cancer drug, we couldn't even measure the binding affinity for. And the only difference between this structure and the cytidine structure is that the 2' hydroxyl is flipped into the other epimeric position. So we wanted to see structurally what was causing this behavior, so we solved the crystal structure of VCCNT bound to gemcitabine, shown over here in purple, aligned with the uridine bound structure in gray. And we found that the binding site rearranges just very slightly. It has to kind of expand just a little bit to accommodate for that added fluorine atom on the other epimeric position, including phenylalanine 366, which we just saw the importance of in a couple slides ago. And TM7B is bumped out a little bit too, and we saw how important it was for interaction with the ribose. Additionally, the ribose of gemcitabine is reoriented with respect to, in gray here are 5 other pyrimidine-bound CNT structures aligned with the gemcitabine-bound structure in pink. And you see how well the other pyrimidine structures align, but the ribose has to reorient in the gemcitabine-bound structure. And we think that all of these slight movements combined could account for the significant loss of binding affinity for 2' substituted drugs. So to summarize the specificity determinants of VCCNT, we saw that it utilizes a bowl-shaped binding site with phenylalanine 366 serving as its base, the base of the bowl, and the ribose interactions localized to one side of the bowl and the nucleobase interactions localized to the other side of the bowl. And we saw that the ribose interactions are extremely important as modification or removal of hydroxyls can result in a significant loss of binding affinity. But there's a little bit more room for modification of the nucleobase interactions that can either increase or decrease binding affinity. So we wanted to see if we could take this information that we gained in these studies and design or improve upon the binding affinity of an existing anti-cancer compound for VCCNT. So what we did is we looked at the structure of pyrrolocytidine, which has the best binding affinity of any of the compounds that we studied. And the only difference between this structure and cytidine is that it has 3 additional carbons here forming this methylpyrrole ring. And since gemcitabine is a cytidine analog, what we did is we just synthesized a compound with the help of Jiang Hong's lab in chemistry that had the gemcitabine scaffold with 3 additional carbons forming that methylpyrrole ring here. So we call it pyrrologemcitabine, and found the binding affinity for VCCNT to be significantly enhanced. So we wanted to see structurally why this was happening. So we solved the crystal structure of VCCNT bound to pyrrolocytidine. And you see that structure up here in this figure. And it adopts a similar binding mode as all the other nucleosides. And the additional carbons that form the methylpyrrole group are shown here in green. But when we look at the surface representation of the transporter shown here, and kind of reorient just a little bit so we're looking almost down the plane of the pyrimidine base, you see that those 3 additional carbons that form the methylpyrrole group slide very nicely into this pocket formed here in between TM6 and TM4, which we call the nuclear pocket, and we think that that accounts for the enhanced binding affinity of this type of compound. So we next wanted to see what effect our modified compound had on the transportability by human CNTs. So we express human CNTs in Xenopus laevis oocytes, or frog eggs. And what you can do with these oocytes is you can measure the current that's generated by ion movement across the membrane upon certain stimuli. And since CNTs are sodium-coupled transporters, they generate an inward sodium current when nucleoside is entering the cell. So we can measure the current that's generated by a method called two-electrode voltage clamp electrophysiology. And shown here are current traces for this experiment. So when you apply gemcitabine to the outside of the oocytes, we see a significant sodium current generated. But when you apply the modified compound to the oocytes, we see almost no current. However, when you add a combination of both gemcitabine and the modified gemcitabine together, we see about a half reduction in total charge uptake, suggesting that the modified compound is binding to the transporter, but it's not able to transport. In HCNT1 oocyte, especially in oocytes on the other hand, we see a little bit of current generated when you apply gemcitabine, but when you apply the modified gemcitabine, we see significantly more total charge uptake, almost 7-fold higher for the modified compound than for gemcitabine. So to see structurally what could be leading to this phenomenon, we modeled the nuclear pocket structures of HCNT3 and HCNT1 onto the VCCNT structure. And looking at the HCNT3 nuclear pocket, it's nearly identical to the VCCNT nuclear pocket. So the methylpyral ring fits nicely and snugly into that nuclear pocket. But with the HCNT1 modeled nuclear pocket, you see that it has a more open conformation leading to the cytoplasm, and it has this bulkier leucine residue here that would actually sterically clash with the methylpyral ring in this orientation, so it couldn't even bind in this mode. It would be bumped out. So if we go back and look at the inward occluded and inward open states of the transporter and the alternating axis mechanism, what we learned earlier is that the VCCNT structure represents an inward occluded state of the transporter, and that's partially mediated by TM6. The intracellular gate is partially formed by TM6. So we think that this type of compound stabilizes this inward occluded state. It binds nicely and snugly here and stabilizes inward occluded state so that it's more difficult to transition into the inward open state for efficient nucleoside transport. But in the HCNT1 structure, we think that it destabilizes the inward occluded state and facilitates the transition to the inward open state so that it's transported more efficiently. And just to test our theory that these are the residues that are important for leading to the subtype specificity, we mutated the nuclear pocket structures of HCNT3 to look more like HCNT1 with just these two mutations, and see significant current generation with the modified compound when we saw none before. And then when you mutate the HCNT1 nuclear pocket to look like HCNT3, we see a reduction in the transportability of the modified compound. So what can we take away from these studies? We found that a combination of both crystallographic and equilibrium binding studies of all the different interactions between transporter and substrate can be combined to better understand the molecular origins of selectivity for that transporter. And the studies also provide proof of principle for using these methods to enhance the transportability of existing therapeutic compounds, and also utilizing slight differences between subtypes for enhancing the selectivity of these compounds. We don't know the clinical effects of the modified gemcitabine, but we do think that this provides proof of principle for using this type of method for improving transport and selectivity of existing compounds. Finally, I just want to briefly talk a little bit about the role of sodium in the ion-coupled transport of nucleosides. So, as we saw earlier, the sodium and nucleoside binding sites are near one another, but the sodium and nucleoside don't directly interact. And over here on the right is a cartoon simplification of the interactions between sodium and the transporter and nucleoside in the transporter. And you can see that they share these two structural elements in common, TM4 and HP1. And we learned through the binding studies and the crystallographic studies that I was showing earlier that several of the interactions with the nucleoside and HP1 are extremely important for nucleoside recognition. So what we think sodium does is it comes in first and binds to its site and pulls these two regions together and stabilizes them so that the nucleoside binding site can be completely formed and form a higher affinity state of the nucleoside binding site. So this theory would predict that in the absence of sodium, nucleoside binding would be very poor. So we measured the KD of VCCNT for uridine using ITC in potassium chloride conditions and saw a significant loss of binding affinity to about 2 millimolar. But it was hard to really efficiently or accurately measure this value because the heats were so low. While in sodium chloride, we get a 45 micromolar KD. Additionally, if you perturb the sodium binding site by mutation, so we mutated the side chain residues that interact with, or that coordinate the sodium, even in the presence of sodium chloride, we see a significant loss of binding affinity comparable to what we saw in potassium-only conditions, suggesting that sodium has to be bound in order for efficient nucleoside binding to occur. We also reconstituted these mutant transporters into proteoliposomes and measured nucleoside uptake in the presence of sodium gradients. And shown over here, we can see that even in the presence of sodium gradients, we get almost no nucleoside uptake by the transporters relative to no gradient conditions. Some other sodium-coupled transporters have been previously shown to be able to utilize lithium for substrate binding and transport. So we wanted to see whether our transporter could do that. So we did the ITC experiment in lithium chloride and saw this is still a very significant loss of binding affinity to about a millimolar as compared to 45 micromolar in sodium chloride. And also measured nucleoside uptake in proteoliposomes with a lithium gradient and see that there's almost no transport relative to gradient-free conditions. So lithium cannot substitute for sodium in uridine binding or transport. So using this information, we developed a hypothetical structural basis for sodium coupling mechanism. And so what we think happens is that in the outward-facing state of the transporter, the sodium and nucleoside binding sites are exposed to high extracellular sodium concentrations that favor sodium binding. And once sodium is bound, it enhances the affinity for nucleoside by completing the formation of the nucleoside binding site so that nucleoside capture can occur. And then upon transition into the inward-facing state of the transporter, the binding sites are exposed to the lower intracellular sodium concentration that favors sodium release into the cytoplasm. And once sodium is gone, the nucleoside binding site structure is destabilized and the affinity for nucleoside is significantly decreased so that nucleoside can be released into the cytoplasm. So over the years, I've spent a lot of time and effort trying to crystallize the sodium and nucleoside-free state of the transporter. But VCCNT prefers to crystallize only in a very specific set of conditions. And those conditions always have to contain both sodium and nucleoside. So in order to circumvent this issue, we've taken a few different routes. One of which is the generation of monoclonal antibodies against the transporter for co-crystallization experiments with fab fragments from the antibodies. And we were able to generate VCCNT-specific antibodies and generate fab fragments that actually bound to the transporter. You see in this figure a size exclusion trace for the complex that runs at a larger size than the transporter or the fab alone. But we've only been able to obtain very small crystals that are hard to reproduce in complex with fab fragments so far. But we haven't yet given up on this method, as we have several different hybridoma cell lines in our hands that we haven't taken all the way through to the fab fragment stage that we will revisit in the near future and try to co-crystallize with our transporter. We've also recently, at the end of last year, we just begun raising antibodies against our transporter, which are single-chain antibodies that are smaller and more compact than fab fragments, and they're often conformationally specific for their targets. So we're hoping that we're going to be able to find a conformationally specific nanobody that can facilitate the crystallization of a different conformation of the transporter. And then most recently, most of my efforts have been devoted to crystallizing a different bacterial CNT ortholog. There's precedent for sodium-coupled transporters from different bacterial sources preferring to crystallize in different conformations. So we figured we would go after another ortholog and see if it was more stable under certain conditions that could favor crystallization of an apostatic transporter. And we've actually found a couple of new constructs that we can get to crystallize both in conditions that have sodium but no nucleoside, and completely apo conditions, so no nucleoside, no sodium. And recently, one of these apo crystals, we were able to collect data at 4 angstroms, which for a membrane protein is getting close to where we want to be. So we're hoping that we're going to be able to get a crystal structure very soon that will, at the very least, we expect to see some local conformational changes around the sodium and nucleoside binding sites, but it could even adopt an outward-facing conformation that's very different from our inward-facing crystal structure of BCCNT that we already have. In the future, we'd also like to ascertain the sodium to nucleoside coupling ratio, so we know whether we should be looking in the bacterial transporter structure for additional sodium binding sites, and also determine the order of binding of nucleoside and sodium using kinetic measurements. Our thermodynamic experiments predict that sodium must bind first for nucleoside binding to occur, but it would be useful to be able to show that kinetically. Finally, we want to measure the nucleoside binding affinities for both the inward and outward facing states of the transporter. A lot of our structural hypotheses kind of hinge on the fact that we think that the binding affinities for the outward and inward facing states in the presence of sodium are going to be similar. So we would like, one method that we're thinking about doing is conformationally locking the transporter via cysteine double mutant disulfide bonds, intramolecular disulfide bonds. So if we can find 2 residues that are close together in the outward-facing conformation but fall apart in the inward-facing conformation, we can mutate those to cysteines so that they lock it in the outward-facing conformation, and then measure the binding affinity by either our fluorescence competition assay or ITC. So with that, I would like to acknowledge first and foremost Suk Young, who has just been the best mentor I could ask for over the years. You know, as you said, we started in the lab at the same time, and he was right there next to me, teaching me all these difficult techniques for membrane proteins, and very hands-on, and has been extremely instrumental in my development as a scientist. I'd like to acknowledge all the members of the Lee Lab, which has grown considerably since I got here, and particularly a couple of former members, John Chung, who participated in part of the BCCNT crystallization, and Junho Lee, who performed the electrophysiology experiments in oocytes. I'd also like to thank my dissertation committee, who has provided invaluable guidance over the years, as well as Jiang Hong and his group for synthesizing that compound for us. I'd like to thank Peugeot, Dick Brennan, and Maria Schumacher for access to their ITC machines. Kyle Daniels for helping to develop that fluorescence competition assay. Charlie Pimble and Nate Nicely and the staffs at AnyCat and SIRCAT for assistance with synchrotron data collection. Bob Lefkowitz and Arun Shukla for technical support with the radio-labeled uptake assay, and our R01 grant for the CNT project, which has funded a lot of the later work that I've gone through. I would be remiss not to acknowledge my friends that I made here at Duke and in Durham, who helped to keep me sane outside of the lab during trying times. And I'm sure that the bonds that we formed here are going to be carried with us for the rest of our lives. And most importantly, I'd like to thank my family for just always teaching me that I can do whatever I set my mind to, and I can be whoever I want to be, and for really just making me the man that I am today. So with that, I'd be happy to take any of your questions.
Fantastic story.
Thank you.
any information on the relative abundance and natural abundance of human CNT 1 and 3?
Yes, yeah, so,
What's the more prevalent?
oh, which one's more prevalent?
Yeah.
I think HCNT1 is more prevalent. But they're very tissue specific, and their distributions.
Yeah. So that relates probably to the FSCS.
Excuse me?
That's most what.
This is just the different tissue distributions they're found in. I mean, some of these are more broadly found, but these are the tissue's highest levels of expression.
So, I have a question actually about this slide. So, you say that CNT3 human is the most similar to the Vibrio, 91% in one copy. You have a stoichiometry of 2 sodiums to 1 base. So, do you think in your structure that's, I mean, you see 1 sodium. Basically, I'm reading a question of, do you think that even the little protein binds 2 sodiums or 1 sodium? And if it only binds 1 sodium, why do you think there's a difference?
Well, it's possible, but the high sequence identity areas that we're looking at are the nucleoside binding site and the sodium binding site. So we don't know what the identity is in the other binding site, the other potential binding site. So it could be completely different residues between BCCNT and human CNT3. So yeah, we've looked all over for different sodium binding sites and looked at some symmetry-related positions since there's that twofold pseudo-symmetry, and looked at kind of the corresponding area of the other pseudo-symmetric position.